Good morning. This is the Oakmont Sunday Symposium, April 11th. And our speaker today is Dr. John Newman, who is fresh from the Buck Institute and going to tell us a little bit about aging into the future with gusto. So quite a topic. Next week, we're going to have Dr. Wen Chen, who's going to talk about human rights in China, and in particular, how the Chinese deal with the various dissident and minority populations there. I won't say it'll be an uplifting talk, but it will be an interesting talk at that. So with that as an intro, Sue, would you like to tell us about John and his talk today?
Sure. Dr. Newman is a geriatrician who cares for hospitalized older adults at UCSF and the VA Medical Center. He focuses on preserving mobility and preventing delirium. His undergraduate education was at Yale. He completed his MD, PhD at the University of Washington and his residency at UCSF. He's an assistant professor at the Buck Institute for Research in Aging in the Division of Geriatrics at the UCSF. He wants to translate our understanding of aging biology to improve the care and help maintain independence of older adults, all of which we want to do. Anyway, without further ado, Dr. Newman, you're on.
Thanks, Sue. So I'm going to share my slides here. And good. Hopefully everyone can see my slides and also see and hear me.
We are not seeing the slides yet, John.
Oh, because I didn't actually share my screen. Sorry. How many times you do this? Okay.
There we go.
That's better. So, hi, everyone. Thanks for that nice introduction. And we're going to talk this morning about towards the future of aging, the future of geroscience, integrating what we're learning about the basic biology of how and why we age into better understanding the problems of older adults and coming up with new treatments for them. So briefly about me, as Sue said, I'm a geriatrician. So I'm a physician specializing in the care of older adults. And I work mostly at the VA hospital and also at UCSF. But most of the time, I'm a basic scientist. I run a laboratory at the Buck Institute for Research on Aging in Nevada, where we study basic mechanisms of aging in test tubes and in mice. But the theme of this talk is going to be how I'm trying to take that knowledge and use it in patients to better understand complex problems of older adults. I'm just going to get my laser pointer going here. So the talk will be in three parts. First, I'll give you an overview of aging biology, of this, maybe a little bit crazy sounding idea that we can actually understand the molecular mechanisms, the biochemistry and cellular and molecular biology of how and why we age. And then we'll talk about how as the biology is maturing, how we're translating this knowledge into clinical studies and clinical trials. And then finally, I want to give you an insider's look of what it means to really be in this field through my own career. So what do I actually do? How do I integrate this basic science at the Buck with clinical studies at UCSF and elsewhere? What does it mean to be a geroscientist with a particular focus on my research around delirium, which we were chatting a little bit about before the talk. So first, aging biology. What's the promise of understanding aging? Heart disease is a huge problem. It causes a lot of death, a lot of disability. We understand a lot about what causes heart disease. And we know how important it is to manage your blood pressure, to stop smoking, to lower your cholesterol, to not get diabetes or to treat diabetes if you have it. And all of these things are very important. Each of them can increase your risk, and if you treat them, can lower your risk of heart disease by 60, 70, 80%. And by carefully addressing all of these risk factors, and hopefully you are all talking to your doctors about these risk factors from time to time, by carefully addressing all of these risk factors, we've made a huge impact on the effects of heart disease on all of us. Heart disease is less likely to kill you than it was 20 years ago. It's less likely to make you disabled than 20 years ago. It's been a gigantic success story. And yet, all of these important factors, blood pressure, smoking, cholesterol, diabetes, if you put them next to the risk of heart disease generated by aging, they almost disappear. Aging is by far the dominant risk factor for heart disease and for so many other chronic diseases too. We call these actually aging-related diseases. Diabetes and heart disease and strokes and Alzheimer's disease and Parkinson's and COPD, and just go on and on and on and on. Aging is the dominant risk factor. It's so dominant that we sometimes don't even think about it because we assume we can't do anything about it. But if we could understand the mechanisms by which aging generates this risk of disease, if we could lower this risk, even by a small fraction, it would be better than all of the other things we do for heart disease combined. So this is one of the promises of studying aging. If we can reduce these aging-related risks just a little bit, we could have a huge impact on helping people to live healthier longer. The other promise of aging is that it's never just one thing. Because aging is the primary risk factor for all of these different diseases, you don't tend to just get one of them. They tend to run together. So it's unusual to see someone who only has heart disease. They have heart disease, some chronic kidney disease, some diabetes, maybe some other vascular disease. This is called multimorbidity, the co-occurrence of several aging-related chronic conditions. And as we get older, not only does the risk of all of these individual diseases go up, but so does the risk of having several of them. And this risk goes up exponentially. So by targeting aging, by targeting the common risk factor that's driving all of these, we hope that not only can we lower the risk of heart disease, for example, but the same intervention, the same drug, the same treatment might lower the risk of all sorts of diseases all at the same time and have an additive effect on helping to keep you healthier longer. Another aspect of aging that is native language to a geriatrician, but if you're not experiencing it, is kind of hard to see, is the concept of geriatric syndromes. These are complex, multifactorial problems that are not like traditional diseases. They don't just have one cause. They have lots of causes all together. Lots of causes spread across all the different systems of your body, all of which come together to create this integrative outcome that has a huge impact on your health. So a fall is the classic geriatric syndrome. Why do people fall? If you ask someone, they might say, I tripped on a curb, or I stepped on something, but that's not why they fell. It's because of the vision problems. It's because of maybe the Alzheimer's disease, maybe just the heart condition, maybe the pills that make them a little bit lightheaded. Maybe it's the weakness in their muscles. It's the tremor. It's the wrong cane that they're using. It's all of these things together that result in a fall, or maybe result in not being able to prevent a fall. It's a complex multifactorial problem. These geriatric syndromes like frailty, cognitive decline, mobility decline, delirium, these have huge impacts on people's lives and are very challenging to treat because they have many different causes, which are different from person to person. But almost all of those causes are driven by aging. So by understanding the mechanisms of aging, we might be able to get in at the ground floor of this seemingly complex, intractable problem and effectively prevent it and treat it. This is the focus of my career, is trying to use the biology of aging specifically to better understand and treat geriatric syndromes. And we'll talk a bunch about delirium in a little bit. Most of us want to live a long and healthy life. The reality is that most of us will experience a period of illness and disability towards the end of our life. When we think about how we're all living longer these days, most of us hope that that's going to be in good health, but fear that it's not. that, in living longer, we may just be extending a period of illness and disability where we're not able to do the things that we want to do because of health problems. Our hope, though, is that in targeting aging, we won't just live longer, but by delaying multiple chronic diseases, by delaying geriatric syndromes, we'll extend the healthy years of life. This is called health span, and is a kind of key concept in the field of geroscience, that we don't want to just extend lifespan, we want to extend health span. If that winds up extending lifespan too, that's great, that's a bonus. And the good news is that what we've learned in the laboratory is that when you do intervene to treat aging, you tend to not only extend the healthy part of an organism's life, but you make it live longer too, longer and healthier. So this is our hope, to extend health span. And in doing so, maybe we'll extend how long we live too. Sorry about that. So how do you actually study aging? This sounds like kind of a crazy idea, but I'll try to convince you that it's really not unlike studying any other problem in medicine or biology. First, you have to describe it. What is aging? What do you know when you see it, but how would you put that into words?
Hello, this is Walgreens, located at the Northwest.
Sorry, you have to listen to my Walgreens voicemail while I talk.
Calling to let you know your
Aging might be described as a gradual, progressive, universal loss of function that begins not long after we stop growing. But you can describe aging, right? Once you've described it, you can more precisely define it. You can define it in ways that you might be able to measure. So, for example, aging is susceptibility to disease. We talked about how aging is the major risk factor for all sorts of chronic diseases. Aging is, unfortunately, about the increasing probability of dying. Aging involves the loss of resilience to stressors. The same problem that affects a younger person will affect an older person more severely. That's not because of how many pages have turned on the calendar. That's because of changes in aging biology. And aging can also be defined as loss of reproduction capacity. And in fact, changes in reproduction are one of the youngest changes that happen with aging, particularly to women. So once you've defined a problem, then you can operationalize this definition to do science, to do experiments in the laboratory, to do clinical trials with people. So for example, you might, thinking about reproductive capacity, you could see how many times a cell growing in a test tube can divide. You could see how long an organism lives. You could see how many chronic diseases a person accumulates. And then you can use these very specific definitions to do science. So having kind of defined aging in a way that lets you do science with it, you can apply all of the high technology that we've developed to study biology in the last few decades, and the sorts of technology that we're using to study cancer and Alzheimer's disease, and use it to study aging too. Model organisms, transgenics, sophisticated genetic manipulations, high technology, high throughput automated systems. We'll talk a little bit about this. These have all been applied to aging in the same way that they've been applied in the past to cancer, Alzheimer's disease, and others, and are leading to breakthroughs. I want to tell you a bit about worms. We study aging in worms at the Buck Institute, and that seems crazy. What could a worm possibly tell us about how we as human beings age? It turns out a lot. So how do you study aging in worms? So let's start with that general description we used, a gradual progressive loss of function. Do worms show that? They do. Maybe we'll define aging as increasing probability of death. And that means trying to study how long an organism lives. We can measure the lifespan of a laboratory worm. Worms only live a few weeks, so it's a lot easier to do experiments in a worm that test their lifespan than it is to do the same thing in a mouse that lives for 3 years, or in people who live for 80 or 100 years. Can you tell what an old worm is? This is a picture of 3 worms of different ages. There's a young worm, a middle-aged worm, and an old worm. And we'll see if I can play the video. Let's see. There we go. Hopefully you can see the worms moving, or some of the worms moving. And you can probably tell which worm is the young worm, which worm is the middle-aged worm, and which worm is the older worm. Just by how they look, and even more importantly, by how they move. So the young worm, of course, is the one that scooted right up off the page. The middle-aged worm is the one that moved less, and the older worm is the one that didn't move much at all. Worms visibly age just like people do. But by studying aging in worms instead of people, you can do things that we would never want to do in people and couldn't do in people, like genetic screens. So we can take thousands of genetically identical worms, zap them with something to create random genetic mutations in them. And now we have a whole bunch of worms that all have 1 or 2 tiny genetic differences from each other. And then we test them. We do some sort of assay, some sort of measurement. For studying aging, we can actually measure their lifespan. You can measure the lifespan of thousands and thousands of different worms. And then you can see, which of, in general, worms with mutations probably won't live as long. Most mutations are harmful. But occasionally you'll find a worm whose mutation makes it live much longer. Cynthia Kenyon, when she was working at UCSF about 25 years ago, did this. She did a genetic screen looking for genes that affected aging in worms, and she found one, actually found several. She found one called DAF2 that made worms live more than twice as long. So this is the lifespan of a normal worm. All the worms die within a month. The average lifespan is about 3 weeks. But when they had this mutation, they lived 2.5, almost 3 times as long. A single gene mutation made them live several times longer. And it wasn't just one mutation. She found several genes that all interacted together. Some of them extended lifespan by themselves, but not in combination. Some of them kind of offset the effects of others. She mapped out a whole pathway, a whole lifespan pathway in worms. This is super cool. Does it have any relevance at all to people? Well, years later, when we were finally able to sequence these genes and find out what they were, it turned out that this was the worm version of the insulin and growth hormone pathway. The very first gene she discovered was the insulin receptor. And of course, this is hugely relevant to people. And in fact, when you look at what gene variants centenarians have, people who make it to 95 or 100 independent and in good health, the most common gene variants are in the insulin and growth factor signaling pathways. The genetics of longevity in worms are the same as the genetics of longevity in people. This has really helped us to understand the basic science of aging. And now we can when Cynthia Kenyon did these experiments, she was looking through a microscope at one worm at a time. But these days we can apply the advances in computer revolution and automated systems and artificial intelligence to do these sorts of things faster and in higher throughput than we could ever have imagined. So at the Buck Institute, we have a worm lifespan machine. This is a whole rack full of scanning machines into which you can place dozens of plates with thousands of worms. There are dozens of these machines. Their movements are video tracked constantly throughout their weeks of lifespan, processed through a computer system that can tell you how much they move and when they're alive and when they die. And so we can do automated high throughput lifespan screening of thousands of different genes, of hundreds of different drugs all at the same time. Other researchers, a researcher I collaborate with at UCSF does the same thing with yeast. Yeast, it turns out, is a great organism for studying the genetics of longevity. And he has a high throughput microfluidic yeast lifespan machine that can follow the life of a single yeast cell, but do this for thousands of yeast cells at the same time. So we can really learn our knowledge of the genetics of aging, but also of interventions that target aging is growing exponentially as we're applying this high throughput science to it. And this has all led to a lot of understanding about the molecular mechanisms that drive aging. You can put these into kind of a nice organization that we often call the hallmarks of aging. And you've probably heard of some of these, things like telomeres, like mitochondria, cellular senescence, like we'll talk about more in a moment, stem cells, inflammation. These are molecular hallmarks of aging, the processes that drive aging. To understand these, I put them into a couple of categories. So first is damage. So damage is just a fact of life. As we live, our genes get mutated, our mitochondria generate reactive oxygen species, which damage proteins and damage fats. We accumulate epigenetic damage, our telomeres shrink. Damage is just a part of life, of all life. And it's part of aging as damage accumulates. As this damage accumulates, though, it's not just a problem itself, but it causes bigger picture problems too. So if you get enough mutations in a mitochondria, that mitochondria stops producing energy for the cell. If you get enough mutations in a stem cell, it can become a cancer. So you can get these big picture consequences that are sort of epiphenomenon from damage. But there's an upside too. So this is all bad. But the third category are the pathways that all of our cells, that all organisms have, that help to repair damage and help to clean up or stop the consequences. And so these are the three categories of mechanisms that drive aging: damage, the bigger picture consequences of damage, and then the pathways that cope with that damage and prevent the consequences. And there's a knife and fork there in the little icon for preventing damage. And this is because many of the pathways that help to clean up damage and prevent consequences, these stress responses, are actually signaled by nutrition. So our bodies have these endogenous, these built-in stress responses, which in many cases come from, in kind of deep evolutionary history, this trade-off that organisms would have to make between surviving and growing. And if there's lots of nutrition around, our cells, a worm wants to grow. It wants to grow and divide, create offspring, make bigger muscles, that sort of thing. If there's not a lot of nutrition around, it wants to survive until a better time. And so it hunkers down and it turns on all these cellular pathways that help to repair DNA damage, help to repair oxidative damage, that help the organism to survive longer. And it turns out that repair mode is good for aging. Now, everything has a time and a place. You wouldn't want to be in just one mode or the other all the time. We need muscles, for example. But more often being in a repair mode, it turns out, is good for aging healthier longer. And this is where, if you guys have heard the relationship between dietary restriction or fasting or eating less and things like that with aging, this is the molecular basis for that. The good news is, though, that it's not about the eating less. It's actually a very sophisticated process in which eating less or eating less of certain things activates molecular sensors in your cells. We've learned a lot about exactly what these sensors are, and we can manipulate them directly. And in fact, many of the breakthroughs in aging science that have led to the first generation of drugs in clinical trials come from understanding these energy and nutrient sensing signals that in turn turn on stress responses, turn on repair. My own lab studies one example of this, ketone bodies, which are molecules that your body makes when you fast, but we're learning has a key role in turning on repair and stress response pathways related to aging. So we're well into the stage where we're not just understanding why we age, but we're creating interventions to target these processes. I want to focus a little bit on a process called cellular senescence, which a number of scientists study at the Buck, and which is also one of the first processes that's leading to drugs in clinical trials. So when your cells get damaged, if they get damaged enough, they'll turn into a cancer cell. But your cells can sense this. If they sense that they've gotten damaged but aren't too far gone yet, they'll turn themselves off. They'll become senescent. They'll turn on a program where they stop dividing so that they won't become a cancer cell. And this is good. This is one of the ways that your body protects you from getting cancer. But senescent cells aren't quite dead. They're sort of like zombie cells. They still release this all these inflammatory molecules, probably to try to get the immune system to finish them off. But the result is that they contribute to inflammation throughout the body and can actually harm the cells and the tissues nearby them. So senescence leads to inflammation. You can kill senescent cells with drugs that specifically target them, called senolytics, lytic for killing and seno for senescent. If you kill senescent cells in the laboratory, you can help keep an organism like a laboratory mouse healthier longer and extend its lifespan. And senolytic drugs are among the first generation of interventions that are entering clinical trials targeting aging. So let's talk about those clinical trials. The big picture here is, as we learn more about the specific mechanisms of aging, creating targeted interventions that hit those mechanisms of aging, like senolytics to target senescent cells, or drugs like metformin to target nutrient signaling, and then use them in clinical trials to target complex problems of aging, like multimorbidity, not just targeting one disease, but targeting a bunch of them that are all driven by aging, or targeting geriatric syndromes, like I talked about earlier, or trying to make people more resilient, trying to give an older adult who breaks their hip the same resilience as a young person who breaks their arm. So this is the scheme for turning biology of aging into clinical trials. And we think about kind of two broad frameworks for clinical trials. One is to try to provide resilience. So for example, you may be taking care of yourself, living independently, playing tennis, but if you fall and break something, that's a problem. That's a problem that you need to recover from. And if you're physiologically, biologically younger, you'll recover faster than if you're biologically older. And we hope that by giving interventions that target aging, we can help make people more resilient and recover faster. Another scheme is targeting health span, which is kind of a longer term thing, trying to keep people healthier for longer and preventing the gradual decline or gradual accumulation of chronic diseases or gradual disability that can happen as we age. So resilience, a short-term thing, coping with a stress, or health span, a longer-term thing, staying healthier longer. These may sound like innovative, new, slightly crazy ideas targeting aging and being healthier longer. Geriatricians already do this. This is how we do clinical trials already. We just don't generally do them with drugs. So for example, I work in a hospital where there's been a lot of research about how to arrange a hospital system to boost the resilience of older adults. And you can really, really help people. You can help make sure that people who come into the hospital independent leave the hospital independent by doing these specialty care settings. It's not with drugs, but still, these are interventions that target resilience. And exercise. Exercise is the magic aging wonder drug right now. And there have been a ton of clinical trials studying how exercise programs, or just being more active in general, can prevent multiple chronic diseases, can prevent diabetes, can keep people mobile and keep them strong. So we already do health span studies with exercise. We just are only beginning to do them with drugs. One of the most prominent examples of this is the TAME trial. TAME stands for Targeting Aging with Metformin. This is a large randomized placebo-controlled trial of several thousand people that will last for 3 to 5 years to see if metformin, which is a drug that is already in use for diabetes, but which targets aging mechanisms, will prevent chronic disease. Not just one, but several of them. So TAME is enrolling people who are 65 to 80 years old, people who already have one major disease or are at risk for disease based on their gait speed, a biomarker of aging. And then TAME is going to test if these people are protected from getting a whole basket of aging-related chronic diseases, including heart attacks, strokes, heart failure, cancer, dementia, or dying. So, most clinical trials will only try to prevent one thing. The whole idea behind TAME is to see if targeting aging prevents all of these together. And along with studying these big picture outcomes, TAME is also studying disability and will collect data on aging biomarkers to help to accelerate other aging-related clinical trials. TAME is all planned out. It's happening at centers across the country, but it's not quite happening yet. They're hoping to start this study, well, hopefully this year. But in the meantime, other studies are happening. So for example, there have been clinical trials using rapamycin, another drug that targets nutrient signals like metformin, to prevent respiratory infections in frail older adults. There's currently a randomized trial going on to see if it prevents severe COVID disease. Studies that use stem cell infusions to treat frailty. And then in the resilience setting, seeing if rapamycin can improve the response to vaccination or improve rehabilitation in older adults after cardiac surgery, or studies of metformin to see if it improves the outcomes of exercise training in older adults. So these are examples of the first generation of geroscience clinical trials. They're small, they're essentially proof of concept studies. They're mostly using drugs that already exist while new drugs are coming through the pipeline. But these clinical trials are already happening. So in the last 15 minutes or so, I want to kind of build upon that to tell you about what it's like to have a career as a geroscientist, what my lab and I do, and how we integrate this basic science and clinical research in my own favorite and least favorite problem, delirium. So I'll tell you a little bit more about myself first and kind of the scope of my career as a physician scientist. Being a physician scientist is a fascinating career. You can do all sorts of different things. And indeed, all sorts of different things seem very important to do. So on one end, I study the basic science of how ketone bodies affect the aging brain. We do this in my laboratory at the Buck Institute, using test tubes, using cells, and using laboratory mice. I also study delirium in a translational sense. So taking what we know about basic science and trying to understand how that applies to this clinical problem. I'll tell you more about that. One of the core missions of a physician scientist is teaching. And that's especially important in geroscience because I'm so happy to meet you guys as a geriatrician who does this kind of translational work, but there's only about 12 people like me in the entire country. Not nearly enough to do the research and the clinical practice that we need to really advance geroscience. And that's where my teaching and mentoring comes in. A big part of my career is trying to reproduce, try to make more people like me who can carry this research forward. And then of course, at the clinical end, this is all anchored in my experiences as a hospital-based geriatric doctor and seeing the problems of older adults firsthand in the hospital, and both being happy that I have tools to help them, but also really, really wanting to have better tools and trying to then make those myself. So my lab at the Buck Institute, on the basic science side of things, studies how ketone bodies, these small metabolites that your body makes when you fast, to study how they provide resilience to the aging brain and in Alzheimer's disease. So we do some very kind of detailed work about how ketone bodies affect epilepsy-like activity in the brain of mice with Alzheimer's disease, try to understand the molecular mechanisms of what are the genes and proteins that ketone bodies affect to improve memory in these mice. The same with understanding how they can improve memory in normal aging. And then both aging and Alzheimer's disease, as I'll talk about in a moment, are the key risk factors for delirium. And so the other part of my lab studies how ketone bodies and how other aging mechanisms affect how likely someone is to get delirium, and how we might turn some of these into treatments for delirium. So I want to tell you guys a little more detail about delirium. Some of you I know from before the talk have experienced this firsthand. If you haven't seen delirium, it's probably the worst medical problem that you've never heard of. It's been called the epidemic within the pandemic. Let me tell you a little bit about it. So delirium is an acute confusional state. It's a sudden problem that's brought on by some sort of trigger, usually a trigger of being sick, of having an infection, maybe of having a stroke or heart attack, or of medications that affect your brain. But one way or another, there's this trigger that kicks off this serious acute confusion. People don't know where they are. They don't know what's going on. They have hallucinations. These hallucinations can run the whole gamut from, just seeing doors move or seeing faces that aren't there, to really horrific things. Survivors of serious illness who are in the ICU will tell you about these Lovecraftian horrors that their brains played on them. Taking normal, real experiences like a nurse coming to draw blood and turning that into, someone cutting off their arm and drowning in a sea of blood and falling into an abyss, and just absolutely horrific stuff. And these hallucinations are a common component of delirium. Paranoia, often delusional paranoia. In many older adults, you see this focused on family members, which is incredibly stressful for family members. And delirium can either make someone very hyperactive or very sleepy. Either way is bad for them and is bad for their medical care. You can imagine that someone who's both confused and very hyperactive, and they have IVs in their arms and maybe a feeding tube in their nose or a catheter in their bladder. And what is all this stuff? And of course, they try to pull it out, and this causes problems for their care. But the opposite is just as bad. If they're sleepy and not wanting to wake up, not wanting to get out of bed, or not able to get out of bed, not able to interact with people, that's also just as bad, if not worse, for their care. And delirium is a problem not just because this all sounds so horrible in the moment, but it has long-term consequences too. Delirium is generally reversible. It will go away when the trigger is fixed, but it can have long-term consequences. People who get delirious in the hospital are about twice as likely to die over the following year, and up to 10 times as likely to get dementia, Alzheimer's disease, over the coming year. So delirium is a long-term problem too. We know a lot about what the risks are for delirium. And actually, we know quite a bit about how to prevent it too. We know that aging and dementia are the primary risks. And we know that if you do a lot of simple things really carefully, you can prevent a lot of delirium. If you make sure people get out of bed and walk, if you make sure that they can hear and they can see, that they have hearing aids and that they have their glasses, if you let them sleep in a dark, quiet place at night and make sure that they're up and awake during the day, if you give them normal social interaction, keep their brains occupied, these all sound so simple, but if you do them all together, you can prevent about 1/3 of all delirium in the hospital. So right now, this is the cornerstone of delirium prevention. And this is what you can do. If you or a loved one find yourself in the hospital, you can help make sure that they're following these simple best practices, like making sure that you get out of bed and walk. Single most important thing to do in the hospital. I wrote it here like 4 times. Stay out of bed, walk. The most important thing you can do to keep yourself healthy if you find yourself in the hospital. It's okay to ask for quiet and dark at night. It's okay to ask to get up and stay active during the day. It's okay to ask people to slow down. Make sure you know what's going on, what you're getting, what's happening. It helps to surround people with the familiar. Family, friends, caregivers are a critical part of preventing delirium in the hospital. And it's important to be careful with medications, especially medications that affect the brain, pain medicines, sedatives, sleeping medicines, things like that. So these are all things that you can do to help reduce your risk or the risk of a loved one getting delirium in the hospital. But we want to understand it even better and try to eradicate it. So what do we know so far about the science of delirium? Not enough, but some. So we know that inflammation is involved, inflammation that one way or the other gets into the brain, usually through a leaky blood-brain barrier. And your blood-brain barrier gets leaky when you're older or if you're sick. We know that when that inflammation gets into the brain, it affects energy production in mitochondria. Neurons don't get enough energy. And when neurons don't have enough energy, they can't do their jobs. They can't keep up their strong connections to each other. And that causes delirium. This is all reversible. Delirium doesn't kill neurons the way that dementia does. And my lab is studying how ketone bodies by providing energy and by dampening inflammation might be one way to help provide resilience to delirium in older animals and older adults. So we're asking two big questions. Are these mechanisms of aging involved in the pathophysiology of delirium? If delirium is a geriatric syndrome, a syndrome of aging, all of that fun stuff I talked about earlier, about DNA repair, about stem cells, about epigenetics and telomeres, does it matter for delirium? We don't know, and we're going to find out. And then can ketone bodies, as one example of an aging mechanism, provide resilience in delirium? And we're doing this across the whole translational spectrum, from detailed work in the laboratory with mice and cells into patients. And I'm going to tell you a bit about the kind of insider scientist perspective on how each of these elements is useful in attacking a big problem like delirium. So we studied delirium in mice. This probably sounds a little crazy. How can a mouse tell you if it's having hallucinations? But in some ways, mice have similar reactions to being sick as people do. If you simulate an infection in a mouse by injecting it with a piece of a bacteria, you can see that mouse doesn't move as much over the next few hours. And we can measure that. A mouse acts sick. So we can measure this. We can see how when we inject mice with LPS, they move less. And then we can give them interventions, sort of like a mouse clinical trial. In this case, we pre-treated them by feeding them a ketogenic diet for a week before they got their simulated infection. And we can see that they move more. Even better, we can give them a compound that's just ketone bodies, not a ketogenic diet, but using ketone bodies like a drug. And we can see that too causes them to move a little bit more, to have a little bit less of a bad reaction to the simulated infection. So this is one example of what we can do with animals that we can't do with people. We can test interventions much faster and a much wider variety of them. But we can do other things too. We can actually look in the mouse brain then and see exactly what these interventions are doing. What's the state of inflammation in the brain? How much energy is in the brain? We can measure ATP, cellular energy. We can do this in young mice and old mice. We can do this in mouse models of Alzheimer's disease. And we can do genetic manipulations to really understand what's happening. We can make mice that have mutations in genes involved in inflammation or genes involved in ketone bodies, and really understand at a deep level exactly why they're moving less in the simulation of delirium, and how something like ketone bodies are working to help with that. This is work that Wyatt Gray, who's a research assistant recently from Santa Rosa Junior College, but soon going to UCLA, has done in my lab. But still, animals are animals. So animals are great at that kind of very detailed mechanistic understanding. But ultimately, we want to know, does this make any sense in people? So we're working with a colleague, an anesthesiologist at Duke, Miles Berger, who does a clinical study of people with delirium around surgeries. So he studies older adults who have elective surgeries like heart surgery, joint replacements. And he carefully follows them to see if they get delirious, and about 12% of them do. And he collects cerebral spinal fluid from them before and after their surgery. So we're going to look in those cerebral spinal fluid samples for ketone bodies and see if having a lot of ketone bodies before surgery protects them from getting delirium, or if the trajectory of ketone bodies predicts who's going to get delirium or who's going to get more inflammation afterwards. And we're doing this by mass spectrometry. So a postdoc in my lab processes these CSF samples, injects them into a mass spectrometer, has to do some sophisticated chemistry with them to be able to identify a whole bunch of molecules that are very, very similar to each other, but has worked out how to separate these out and quantify them to the micromole. So we can detect very, very small amounts and very small changes. And we're doing this mass spectrometry now on all of these CSF samples to see if there is, in fact, a signal of ketone bodies being relevant in people for preventing delirium. But I want to ask an even bigger picture question too, not just ketone bodies, but aging biology in general. Is this why older people tend to get delirium? And for that, we have to create our own study. So I'm working with Sarah LaHoo, a neurohospitalist, a neurologist at UCSF, who's creating a clinical study around people admitted with hip fractures. Hip fractures are a terribly serious geriatric problem. They're as bad for a frail older adult as a metastatic cancer is. And many of these people get delirium. Even though at UCSF, all those things that I talked about earlier, that the little things you have to do right, we do them all. We're as careful as you can possibly be. We have one of the best delirium programs in the country. And still about 25% of people will get delirium after their hip fracture. So we're enrolling these patients into a study. We'll collect blood from them at several times during their hospitalization. We're going to look at Alzheimer's disease markers and also aging biomarkers. We're going to look at their physiological age. You can measure someone's age based on their epigenetics, based on DNA methylation patterns. So we're going to measure their physiological age and see if that predicts their delirium risk better than their calendar age. And remember, senescence, cells that are damaged enough to stop dividing so that they don't become cancer, but they release lots of inflammation. Well, this might be one of the aspects of aging biology that helps to trigger delirium. So we're going to measure senescence and see if the amount of senescence in a person predicts their delirium risk. And this is going to be a really unique clinical research cohort where, because they're having surgery, we cannot just look at blood, but we'll have biopsies and bone specimens collected during their surgery. So we can really hopefully have a sophisticated understanding of how aging biology in different tissues affects their risk for delirium. And of course, the long-term goal is to figure out new treatments for delirium. So this clinical research cohort can become a clinical trial cohort once we know what we want to test. And finally, the Buck and UCSF are at the center of this. So the Buck Institute is one of a handful of sites around the country that have formed into the Geroscience Network and its educational partner, the Geroscience Education and Training Network. And the whole goal of these national networks, supported by the National Institutes of Health, is to accelerate the progress of geroscience through clinical trials and to accelerate the training and mentoring of new people to do these geroscience clinical trials. And the Buck and me, and we're right at the center of all of this. So that's a geroscientist career in a nutshell. I stay busy, but this is how I try to leverage every bit of the span of my career, from basic science in the laboratory to clinical research, to education and training, to hospital practice, to try to change the care of older adults in one particular way around delirium. And before I take questions, I just want to tell you briefly about the Buck Institute, if you haven't heard. So the Buck Institute is the first and still the largest independent research institute entirely focused on aging biology. It contains everything from, mass spectrometrists to worm biologists to geriatricians, from every aspect of biomedicine, but all brought together by this common mission to target aging. There's about 25 laboratories. These are some of our faculty. And if you may have seen the Buck as you're driving through Nevada on 101. It's that beautiful white building designed by I.M. Pei up on the hillside. There's more than 200 scientists. We've been around for more than 20 years now. We have about 250,000 square foot campus with research labs, lots of teaching and education space, event space. And our budget is more than $50 million a year, almost entirely devoted to science. And some of this comes from the Buck Trust, and some of it comes from philanthropy, but the majority of it comes from NIH grants that we all work to help fund our research. And all of the research I told you about just now is funded by grants from the National Institute on Aging. So aging biology is here. Geroscience, the application of aging biology to human disease, will transform geriatrics. We just need to get there. But we're on our way. Clinical trials are happening now. And it's an exciting time to be in the world of geriatrics and geroscience as we figure out where we're going to go from here. So thank you for your attention. I think we're going to share these slides later. So you're welcome to drop me an email. You're welcome to say hi to me on Twitter. And thank you to all the folks in my lab who work hard to generate the data I showed you and my collaborators. And then most of my work is funded by the National Institute on Aging, and as well as the Buck Institute and some other organizations too. So thanks for your attention, and happy to answer questions.
John, why don't you start by, we'll drop the screen share. Mm-hmm. Okay, with questions, prefer the chat is one way to do it, or if you know how to electronically raise your hand, like Sue Aiken has just done, we can recognize that, but we're not good. There are way too many people on here for us to be able to recognize it if you physically raise your hand. However, being the moderator, I get the first question. John, in a practical sense, many of us in working with our own clinicians recognize they do not have a strong background in geriatrics. How do you recommend we approach very good conventional general practitioners in this world where we're now truly geriatric patients?
Right.
Yeah. And this is even a worse problem in some areas. There's very few geriatricians around. There's only a few thousand of us in the entire country. And I'm talking about scientists, I'm talking about geriatricians, period. Far too few to actually care for all the older adults who would need us. And in some areas, they're even thinner on the ground than that. So there's a fair number of geriatricians in San Francisco. For example, we have great house calls programs, but there are fewer geriatricians in Marin and Sonoma. Ultimately, it's helpful to try to think about how to be your own geriatrician. That's the title of a talk I sometimes do for other audiences, to learn a little bit about how geriatricians think, so that you can then kind of help to walk your own provider through the geriatrician's perspective to problems. And what does that mean? So some things that means are taking a big picture approach to everything. Everything you do should have a goal. Everything medical should have a bigger goal in mind. Not just treating a problem or treating a number for its own sake, but what's the most important goals for you to accomplish, and how can all the elements of your medical care support that? Because sometimes it's about not just knowing what to do, but also knowing what not to do. Because if you try to do everything or try to do too much, you can wind up actually causing more problems than you solve. So a big part of the art of geriatrics is keeping that big picture in mind and being really thoughtful about what are the most important things we need to do to help you reach that big picture. So being very careful with new medicines, for example. One of my mentors taught me that every new symptom in an older adult is a medication side effect until proven otherwise. Trying to get at the underlying causes of things. And this focus on function and mobility. So the most important thing in the world to a geriatrician is how active and mobile and independent you are. Because not only is that usually one of the most important ends to itself, that should be the goal, right? Whatever condition you're treating, the goal is to stay independent and active. But it's also a measure of how well you're doing. So no matter what else you're doing, if you're less mobile, less independent, something's going wrong, and that needs to be fixed. And this is the job description of what I do in the hospital. Even if we're treating pneumonia perfectly, if someone's not able to get out of bed, we're doing it wrong. But the same is true in day-to-day life too. It's really all about watching your mobility, watching your function, watching your independence, and making sure that all of your medical care is focused towards helping to improve that.
Sue Aiken has had her hand up forever. Sue, yep. You're on.
Yeah, okay. Just a couple of quick things. About 10 years ago, I had the pleasure of taking a tour of the Buck Institute, and I'm hoping that you, under normal times, are still doing that, and encourage people to sign up to go on a tour, because that building is phenomenal, including an art exhibit. I mean, it's really an event place in the opening lobby there. I'm one of those that has been taking Metformin for 15 years now, but I'm also 80, so I don't qualify for your study. But is there any interest in studying those who have been on all three of those meds, the cholesterol, the blood pressure and the metformin for about that period of time. Is there any interest in that at all?
Yeah, of course there is. So metformin is widely used. It's still the best drug for diabetes. It's first line, in part because not only is it very safe, but because when it's been studied in diabetes, it has interesting side effects like making people less likely to die.
Good. That's a good thing, yes.
Important side effects. And for a while, it was the only diabetes medicine that had clearly been shown to lower mortality in diabetes. Now there are several others. It's a golden age for diabetes treatment right now. But much of the data that supports testing metformin in aging comes from these studies of people who take it for diabetes. And you know, some of this data is a little bit controversial, that you do one study, you do another study, things are a little bit different, but in general, it looks like when we've studied metformin, as you suggest, that people who take it are less likely to die. We know that. Less likely to get diabetes. We know that. Less likely to have heart problems. We know that really well. Probably less likely to get cancer. Maybe less likely to get Alzheimer's disease. And a really interesting study out of the UK where they tried to compare people with diabetes who take metformin to people without diabetes of the same age and same general health. It suggested that the people with diabetes taking metformin actually lived longer.
Really?
Than even people without diabetes at all. So it's been very interesting to study people who take metformin for diabetes.
Great, thank you.
John, Bonnie asks, are you suggesting that fasting could be useful in preventing dementia?
So from a scientific perspective, the things that happen in your body when you fast may be useful in preventing dementia. And there are studies, and I mentioned how we've kind of narrowed in on molecular mechanisms of this and tried to turn that into, you know, picking out drugs or other treatments. And some of these are in clinical trials for trying to prevent or treat early dementia. Now, fasting itself, I don't think there's a clinical trial of intermittent fasting or time-restricted feeding, as you might say, for preventing dementia. I wouldn't be surprised if there is one. But there's definitely, I think there's strong reason to think that the mechanisms behind this are going to be fruitful. Now, of course, if you talk about what do we know for sure about preventing dementia, it's not nearly enough. And really the only thing with solid, clear evidence is exercise, activity. But the biology of exercise and the biology of fasting are not that different. So there may well be something to what happens in your body when you fast that turns out to be helpful for dementia.
Okay, Laura Lynn, you had a question.
Yes, I do. Thank you. I'm enjoying this very much. Is there a way that we, like local lay people, can get involved in the TAME research?
I guess the thing to say is we're working on it. So that map I showed towards the end with sites around the country for the Geroscience Network, that's also essentially the sites for the TAME study. And you may have noticed the only one on the West Coast was the Buck, and we're not currently a TAME site. So there actually are no sites on the West Coast for the TAME study right now. We're working to change that. And hoping to be able to make UCSF, and in partnership with the Buck, into a site for this in the near future, or similar studies. For the moment, though, I don't think that there are ways for people to get involved remotely in TAME, but there are certainly ways to get involved in other studies around aging.
Through Buck?
For clinical trials, more through UCSF, and actually through CPMC research too. If you Google the San Francisco Coordinating Center, let me see if I can find a simpler URL for you. There's a nice aging number here. drop me an email, and I'll
Okay.
send you a link to a website that summarizes all of the different aging-related clinical studies that are going on around San Francisco, and there's a bunch of them. And if you're interested in, and many of them involve, the biggest one is about exercise and aging, and involves all sorts of interesting, sophisticated imaging studies of your muscle function, mitochondria function, and maybe of interest to folks who really want to get involved in research, but also learn more about their own bodies.
John, would you send Judy or me that, and we'll make sure it's posted on the website.
Okay.
Sure.
Okay.
I have one other minor question, possibly minor. Is there a simple way that I could get, for like layman, ketone measurement? So I know when I'm just lightly, moderately in keto, I'm not going to fast for 20 days or anything, but, just a little, if I can know when I'm in ketosis slightly.
Yeah, well, especially if you're doing some sort of fasting, which many people are trying these days. There's a couple of relatively easy ways to measure your ketone bodies. One is that you can buy ketone urine strips in any pharmacy. These were meant for use by kids, or mostly by kids with type 1 diabetes. But they can tell you if there's some ketones in your urine, and that means that your body is making ketones. It's a very kind of rough and ready sort of measurement. It's not very precise, but it's sort of like yes or no. But in the same way that you can buy a blood sugar monitor where you prick your finger and measure your blood sugar. You can also buy a ketone body monitor. Same thing. You prick your finger, put the strip in the machine, and it tells you exactly what your ketone body levels are. And there's a few companies that make that. They're a little bit harder to find than blood glucose monitors. You probably have to get them. You probably have to order them online, but they're pretty easy to use.
Thank you.
Stephen, you had a question.
Thank you, George. Yes, doctor. I was just curious about, back in the 70s, I went on this and I used keto sticks as part of the program, the Atkins diet, and now there's a paleo diet, which I think is a little bit similar. And I mean, I know some people that went on that about 4 years ago, the paleo, and changed their lives. They're now healthy, active, thinner, happier.
Cooking about then.
I don't know if that's psychological or the diet, but they look great.
Sooner if I get off here.
So could you make any comments about those diets, please? Remember
Sure.
the fork and the knife.
I study ketone bodies in the lab, and I mostly focus on in people, thinking about how we make ketone bodies in normal life. So I don't use ketogenic diets in clinical practice. Other doctors do, of course. But from a science perspective, it's a lot of people have tried ketogenic diets or similar diets. Atkins can be a ketogenic diet. So, I guess let's say, what does a ketogenic diet mean? It means eating a small enough amount of carbohydrates so that your body starts to make ketone bodies instead. So fundamentally, it's about reducing carbohydrate intake a lot. What do you eat instead? Mostly fats. On an Atkins diet, you eat a lot of protein too. You do want to be careful not to overdo protein, because too much protein can be harmful to you, both in general from an aging mechanisms perspective, but also specifically in terms of harm to your kidneys and things like that. So mostly, people who do clinical studies of ketogenic diets, it's really a lot of fat, mostly healthy fats. But there are lots of clinical studies of ketogenic diets in treating obesity and diabetes. There's clinical trials now around Alzheimer's disease, for certain cancers, all sorts of stuff. For kind of day-to-day life, so if you're not talking about starting a ketogenic diet to treat a cancer, but for just day-to-day life, it's interesting that especially if you're inactive and either overweight or with diabetes, in improving getting more active and improving your weight and metabolism is really important to improving your overall health. And we don't know how to predict what diets will help people with that. It seems like people really just need to try different things and find a pattern that works for them, that's sustainable. If some of your friends have found that pattern with paleo or something similar, I think that's good for them. But ultimately, it's about finding a sustainable eating pattern that helps you maintain your weight, but also keep your strength and make you more active.
You mentioned a ketogenic cancer diet. I'm a cancer patient, and I'm just wondering, can you say anything more about that? It's the first I've heard of it.
Well, so this is investigational. So meaning that people are researchers are running clinical trials of medical ketogenic diets for certain specific cancers. The scientific rationale for that is that certain cancers get very dependent on burning glucose for energy, more so than the rest of your body. So a ketogenic diet effectively starves them of glucose, or that's the theory anyway, and may even work better in combination with certain chemotherapies that also target glucose metabolism. So there are certain specific cancers for which this is being studied. If you'd like, you're welcome to send me an email and I can send you some links to what they are. I would be cautious about kind of extending that globally. Like, would ketogenic diet work for any cancer? Who knows? But it's a very good topic for clinical research, and there's rigorous research underway.
John, do you have time for another 3 or 4 questions? We've got a line of them coming in on the chat room.
Yeah, I see. No, I'm happy to stick around and keep answering.
Okay. Practical recommendations to extend a healthy life. If you wanted if you're just sitting here as a normal 65, 70-year-old and you're looking for the 3 or 4 most practical, what's your thoughts?
Stay active and stay connected. The single most important thing that all of us can do to stay healthier as we age, and it doesn't matter what age you are, is to be more active. And not even exercise per se. Like, sure, exercise is great, but just be more active, more mobile, more independent. A little bit at a time. Whatever you can do a little bit more tomorrow, do a little bit more the day after that, and do it systematically. But staying active is 1, 2, 3, 4, 5, the most important thing that you can do to stay healthier. And it's amazing to me just how that gets reflected in every setting. From people who are living in their own homes, but who embark on a program of being a little more active. Call it exercise if you want, but being a little more active, and they're less likely to get frail. They keep their muscle strength. They keep their muscles stronger for longer. They're less likely to get diabetes. They're more likely to stay independent. They're less likely to get dementia. It's like this magic effect of just being more active. And in the hospital, it just blows my mind how the simplest thing, make sure that people get out of bed and walk, because most of the time, no matter how active you are, you go into the hospital, you lie in bed. But just the simple act of getting out of bed and walking a few times a day has such a huge difference in people being able to go home versus to rehab or a nursing home, in preventing delirium. It blows my mind how powerful this simple thing is. So I think you ask any geriatrician, you'll get the same answer. Be more active. And that's the key to staying healthy and staying independent. I would add, stay connected. So just like staying active keeps your body strong, staying connected helps to keep your mind strong. Stay connected to other people. Keep your mind, stay connected to doing meaningful things. And that's also, I think, really important to helping us to age better, longer.
We've got several questions, John, on diet. So let me hit the first one. What are your thoughts on a vegan diet?
I mean, kind of like I was saying earlier, I think the way that most of us eat in modern life is not healthy for us. And it seems like you just need to find whatever pattern of eating works for you. And for many people, that's trying a vegetarian or a vegan diet. And if that works for you, I think that's great. There's some great data about how following a vegan diet can reduce the risk of heart disease, and certainly can help you maintain your weight and your metabolic health. It's bizarre, right, that there's also data that a high-fat ketogenic diet can help to maintain your weight and your metabolic health, but so can a plant-based vegan diet. Hey, and you can do a plant-based low carbohydrate diet too. That works also. But the way that I process all of this is that almost any pattern of eating that's different from our normal is going to be healthier for us than what most people wind up doing by default. There's also certainly arguments about the environmental climate impact of animal meats, and there's an argument for, we should be eating less meat and maybe trying a vegan diet for that too. I admit I like meat. I'm not a vegan. I also don't do a ketogenic diet, so I'm not the right person to ask about any of these.
Okay, last question. In terms of protein, you had mentioned briefly about protein intake. Where do you fall on the, what I would call the grams of protein that you should be taking scale?
And this is different at different stages of life and in different contexts too. And I mean, enough, but not overdoing it. But for older adults, there's actually evidence that having a relatively higher protein intake can help to maintain your muscle strength. So older adults may actually need a little bit more protein per body weight than a younger person does. The molecular rationale for that is that so I've been talking about kind of nutrient sensors throughout this hour, right? So there is a sensor in your muscle cells that senses protein intake. And if it senses a lot of protein intake, it will help to rebuild your muscle strength. If it senses low protein intake, it won't. It'll focus on just keeping your muscles as they are. It turns out that in general, you will do better for most of your life if you're in that repair mode. But older adults especially that sensor gets a little bit sticky, and you need more protein to activate it to maintain the muscle strength you have. This is especially true when you're exercising. If you're rehabbing or doing an exercise program that's focused on building muscle, you have to be really thoughtful about getting protein intake right after your exercise to make sure that signal gets turned on and your exercise turns into muscle. So in terms of, I'm not sure I'd go into a specific number, but dietitians often talk about a gram per kilogram as being a reasonable protein intake. For an older adult, especially if you're trying to maintain your strength, I really would try to hit that. For a younger person, maybe not so much. But the key with different diet patterns, especially low carbohydrate, high protein diets, is not to overdo it too much, not to go too far beyond that gram per kilogram, or that can start to cause harm.
John, I want to thank you on behalf of the Symposium crew here. We are your clients. We are typically quite geriatric. We love the Buck Institute and what it's trying to do. And we hope that you're successful with some of those research programs that you were talking about. As a reminder to everybody, next week our talker will be Dr. Wen Chen, who's going to talk about human rights in China. So she's going to focus on the problems in China for the minorities and the dissidents, and hope to see you then. Thank you again, John.
Thanks, George.
Pleasure.
Thanks, George. Thanks, Doctor.
Take care, everyone.